Okay, well, we're ready to move on to chapter 3, and this is a briefer set of notes. The real focus here is uncovering the very basics of, as the title of the chapter suggests here, the cells of the nervous system. We've already talked briefly about some of the micro level views of the nervous system. We talked about neurons and glial cells in chapter 2, but my mention of them there was relatively brief. So we want to get first into the anatomical structures of the cells and the general types of cells that are found. In the next chapter, we will get more in depth in terms of the physiological processes and the actual activity of the cells of the nervous system. But for now, we're going to start with neurons. So we haven't always known, of course, that the nervous system was made of cells. Even when cells were first discovered in the body, right, that animal bodies are made of discrete cells, it was not clear that this also applied to the nerves. And that's because when you are looking at cells under a microscope, you have to stain them in order to be able to see them, because for the most part, they're transparent, right? You put them under a microscope slide with a bright light, and they are almost impossible to see unless you dye them with a stain so that it makes the cell body and the structures inside the cell more visible. And as it turns out, though, the neurons do not absorb the usual kinds of stains that are used to visualize other kinds of cells in the body. So when people tried to put nerve fibers under microscopes back in the very earliest days of using microscopes to study bodies, it was not really possible to view the individual neurons themselves. And so this idea that the nerve was really a long continuous fiber is something that persisted even after all the other cells of the body had been discovered. In fact, one thing we mentioned back in chapter 1 was that Descartes had this idea that the nerves were really like hollow tubes that were passing fluid like a hydraulic system, moving the cerebrospinal fluid from the ventricles of the brain down into the peripheral nerves and then inflating the muscles to control the movements of the body. But in the 1800s, a guy named Samuel Golgi invented a silver stain that worked well for neurons. And once this was discovered, Santiago Ramon y Cajal was the first to really embark on this long, detailed anatomical analysis of neurons. He was using the Golgi staining technique, and he discovered all of these many different neurons. And so he's the first to report that the nerve fibers were really not continuous long fibers, but were rather discrete individual cells. And he is famous for producing these very detailed drawings. This, of course, is not one of his drawings. These are photographs of, on the left anyway, of the kinds of neural pathways, sorry, that he had discovered, right? So finding that there were these anatomical structures, like we could see a cell body, but all these branching fibers that are parts of discrete cells that connect to other cells, right? And so we learned from this that there is a very standard structure to most neurons. And we see that represented in the drawing on the right, which is again a more modern drawing that shows the cell body or soma that houses the cell nucleus and regulates all of the basic metabolic processes in the cell. And then on one side of the cell body, we find these branching structures called dendrites. That comes from either Greek or Latin, I don't remember, but one of those two. The word, I think it's Greek really, means tree branch. And that's what these, of course, kind of look like, or reminiscent of the branches of a tree. And that's why they are called dendrites. But one of those branches off of the cell body is different. It's called the axon. And we see now a depiction of an axon. And we see that the axon is longer and it has fewer branches on it, except at the very tip of the axon, which we call the axon terminal. And that branches off. And there's also this indicated in this drawing that sometimes axons can be very long, right? And they may travel a long distance within the body. So to some extent, there are some continuous fibers in the body, but those are just extended axons that are traveling to various locations of the body. And so here we have all of that broken down in words here. So again, dendrites, cell bodies, and axons. But here we have their function. And hopefully you remember this from your introductory psychology courses, that the dendrites are essentially the input to the neuron. They are receiving information from other neurons. Specifically, of course, they are receiving information from the axons of other neurons, because as we see at the bottom, the axon is what is the output of the cell. It transmits the information to these other cells. Let me back up for a second here, because there's a little note about dendritic spines, and we can get a little hint of this in both the photograph on the left and the drawing on the right. These tiny little spiky coverings around the dendrites, right? And those are called dendritic spines. And those longer horizontal branches that are coming in, those are the axons of some other neuron, right? But those little tiny little spikes that cover the dendrites are dendritic spines. And the point of this, as it says here, is that we're going to increase the surface area of the dendrite. So later, when we talk about how synaptic function occurs, we may find ourselves concerned with the total amount of surface area on a dendrite, because if we increase the total surface area of a dendrite, that means that we're going to be able to increase the total number of synapses that are able to occur on a given neuron. It also means that we might be able to either increase or decrease the sensitivity of that neuron to incoming information. So increasing surface area increases the sensitivity of that neuron. As noted here, the cell body integrates the information that arrives via the dendrites. And then if that passes a certain threshold, that signal is then transmitted via the axon to the dendrites of other neurons. And that's where we find the synapse. So the synapse here is defined as the connection, junction, or gap between two neurons, between the axon terminal of one neuron, what's usually referred to as the presynaptic neuron, and the dendrites of the postsynaptic neuron. The very tips of the axon terminals are called terminal buttons, because they are little rounded button-shaped endings on the tips of those terminals. And that's where we find the neurotransmitters, right? The actual chemicals that are released into the synapse. Neurotransmitter travels across the synapse to activate receptors found on the dendrites of the postsynaptic cell. So we have a little mapping there of just the information flow on the bottom here. Again, incoming input from the dendrites organized into the cell body, transmitted via the axon, and then across the synapse to the dendrites of another neuron. And so on it goes for across the 80 to 100 billion neurons in the brain. Now, something else that we want to think about is that not all neurons look exactly alike. We tend to have this sort of a stereotypical view of a neuron that we saw a couple of slides back that has its dendrites and an axon. And these pictures usually depict what we call an interneuron, right? Neurons that transmit information between other neurons. But they are not the only kind of neurons. There are also sensory neurons. So a sensory neuron is technically a receptor that receives information from the environment rather than from other neurons. So for example, photoreceptors, which means light receptors, which we typically call the rods and cones that we find in the retina of the eye, those are neurons, but they don't have dendrites and they don't receive input from other neurons. Not really. I mean, there's actually a little exception to this, but primarily the rods and the cones are stimulated by light, and the light triggers a chemical change inside of those cells that then is transmitted via their axons to other cells of the visual system. Likewise, the cells of the inner ear that are used for hearing are hair cells, and when vibrations from sound waves enter into the ear and activate the different moving parts of our ears, this eventually causes a compression of the tips of these hair appendages on these hair cells that then activates an electrical potential in those neurons. So again, they don't receive input by dendrites, they receive input because of a mechanical activation of the hairy appendage coming off of these neurons. or the touch receptors in our skin are considered mechanoreceptors or pressure receptors because they respond to mechanical pressing and stretching of our skin. The taste and smell receptors are chemical receptors, and so they have a kind of a receptor that responds to a particular form of chemicals in foods that we eat or things that we inhale and smell. And that again activates electrical changes in these neurons. So these are all part of the afferent sensory system, right? Bringing that sensory information in. It begins at the receptor surface, right? So the retina of the eye, the hair cells of the inner ear, the mechanical receptors in the skin, the taste buds in the tongue, and so forth. And then that's transmitted by the sensory nerves. So we previously learned in the last chapter about the cranial nerves as well as the spinal nerves. And that's all part of that sensory, somatosensory body input, right? That is called afference. And then it goes from the spine and brainstem up into the relevant cortical areas for further processing, which of course is where we'll find interneurons, right? Neurons in the neocortex are going to be interneurons that communicate with each other. So that's information traveling from neuron to neuron. Now, eventually we may also want to move our bodies in response to the sensory information. And so interneurons in the motor areas of the brain, we talked about the motor cortex, the basal ganglia, cerebellum, and how all of that stuff helps kind of regulate and organize the way in which we perform movements. But eventually this needs to trigger a muscle contraction. And what this means is that the signal descends down from the brain to the spine. This is called efference. And there are motor neurons in the spine. Motor neurons are essentially neurons that have a cell body located within the spine and dendrites located within the spine that are receiving input that's descending from the brain. But their axon travels and synapses onto muscle fibers, right? So the synapse here is not from neuron to neuron, it's from neuron to muscle fiber. And so this is a specialized kind of synapse that directly triggers the muscles to contract, and that's what the motor neurons do. So here are some depictions of, on top, a sensory neuron. The one on top here is for a skin detection touch, right? So we have sensory endings, in this case, these kind of free nerve endings embedded within the dermal layer of the skin, the subdermal layer of the skin, really. And any pressure on the skin will bend and deform or stretch the skin, which then bends and deforms those nerve endings, which is then detected as a signal, right? And that signal then travels into this fiber, activates the cell body. We see the soma there in the middle, and that will then be further transmitted to the spine where it then is relayed up to the brain, right? But that's a particular kind of sensory nerve that is dedicated to processing this touch information. Down on the bottom, we have a motor neuron, right? So in this case, the cell body with its dendrites seen on the left, this would be found typically in the spine. So it's receiving descending information from the brain. And then the axon is actually part of a spinal nerve that leaves the spine via the ventral root. If you remember the dorsal and ventral roots of the spine that we talked about before. So in this case now, the ventral spinal root will provide that motor signal that goes out to the muscle fiber. And we see the synaptic terminals here directly synapsing onto the segments of the muscle fiber itself, causing those individual little segments. That's, you see the stripes in the muscle fiber there. Those stripes are basically dividing different little segments of the muscle. And those presynaptic terminals are synapsing onto those segments and causing them to individually contract, which then results in the entire muscle fiber length contracting. And it's through this rapid contraction of the muscle fiber that shortens the muscle, which then results in movement, right? So if you think about your bicep, if you just do a bicep curl, for example, in order to, when your arm is fully extended, in order to activate the elbow, you have to shorten the bicep muscle. And by shortening the bicep muscle, contracting it, you bring your hand and your forearm up closer to your body, right? We mentioned in the previous chapter that there are glial cells, and I provided a brief explanation for what they do, but here I have a more complete list of the glial cells. So the first ones here are known as the ependymal cells, and these produce cerebrospinal fluid. So what we have in terms of cerebrospinal fluid is really something that needs to be continuously regenerated because we lose it. It leaks out through various parts of the lymph systems in and around the brain and the neck. So that has to be regenerated, and so that's found in the walls of the various ventricles of the brain and spine. Right, these ependymal cells do that. I had mentioned the job of the astrocytes specifically in chapter 2. These are the ones who provide some of the real main life support for the neurons. They constitute the blood-brain barrier. They wrap themselves around the arterial blood supply. They extract nutrients from that and transfer that to the neurons. And then when the neurons generate waste products, they also clean that up and then transport that into the veins so that it is then transferred to the kidneys to be excreted through the urine. Something else that astrocytes also do, and this of course is not something that is a common thing, but in case there is damage to a nerve, you may have heard the term before of what's known as a glial scar. This is one of the reasons why healing nerve injuries is so problematic. So if you sever a nerve, that means you have severed perhaps some axons. And the trick, of course, is how do we heal that? How can we possibly reconnect those axons? That's not so easy to do. Or maybe do we want to regrow axons to bypass that? That's, again, not so easy to do. And part of what also happens here is that the astrocytes detect that injury and they form additional new astrocytes all around that injury. And essentially they cover up the ends of those broken nerve fibers. This is now what's called the glial scar. And that actually, in a sense, is something that presents a barrier to healing in a way, right? Because it's harder to regrow an axon there if the glial scar is actually blocking that gap, that severing point in the nerve. Here is a picture of an astrocyte doing its job as part of the blood-brain barrier. So again, we can see how the astrocyte has appendages that connect to the blood vessel. The blood vessel is shown here as having so-called tight junctions. And the idea here is that the arteries that feed the brain, normally the capillaries, will directly feed cells by leaking a little bit of the oxygenated and glucose-rich blood to those cells. But here, these tight junctions prevent any leaking of that. So that way, none of the blood supply comes into direct contact with neurons. Instead, the astrocytes have these appendages that wrap around those junctions and extract nutrients directly from the blood supply and transfer it directly into the neuron. As you can see, it also has appendages that wrap themselves around different parts of the neuron to supply that. So the astrocyte here is literally the blood-brain barrier. It is the intermediary between the arterial blood supply and the neuron itself. Now we have a couple of others. The microglia. These are part of the cleaning system of the nervous system. So just as I said with the astrocytes doing some job of cleaning things up and transferring that to the veins, microglia do the same thing as well, right? So they kind of work like the immune system, right? They get rid of things that we don't need in our body. And what microglia do in particular is they eat dead cells, called phagocytosis, right? So any neuron that dies, for example, we might want to clean that up and get that extracellular material out of the brain or nervous system. And that happens in development sometimes in an automatic way. It's just a normal part of early brain development. It's called apoptosis, where some of our extraneous neurons end up dying off. But even throughout the rest of our lives, this can occasionally happen in different ways. And so microglia do their job to help clean that debris away. Oligodendrocytes and oligodendrocytes and Schwann cells kind of do the same thing, right? These are the particular glial cells that generate the myelin sheath. And we'll talk a little more about the myelin sheath later. It surrounds the axon and helps speed up the nerve transmission process. But we make a distinction between the myelinating cells of the central nervous system, which are the oligodendrocytes, and the myelinating cells of the peripheral nervous system, which are the Schwann cells, right? I also might have mentioned in the previous chapter that during early brain development, during the embryonic and fetal stages of development, the neurons need to know where to go in order to start forming and giving structure to the brain. As it turns out, most of the neurons are actually born within the spinal column, right? So in the spinal cord, there's a layer in there called the mantle layer, which is where neurons are created. This process is called neurogenesis, right? So very rapidly, billions and billions of neurons are being made in the course of our 38 weeks of prenatal development. And so we're born with nearly all of those 80 to 100 billion neurons. But because they're grown in the mantle layer of the spinal cord, that means they have to migrate to the appropriate location as the limbs start to form. For example, the nerves grow out with it to form the nerves, the spinal nerves that innervate the muscles and the skin of our arms and our legs. And as the brain is developing, of course, then other neurons have to migrate up the spine towards the brain to start forming all of the cerebral cortex and all the other parts of the brain. So this process is called neural migration. But in order for the neurons to migrate correctly to get to the appropriate location so that the brain and the nervous system grows and you get the typical shape of the brain that we are supposed to have, that's the job of the radial glia. The radial glia essentially form a lattice work or a kind of a map that these immature, brand new neural cells migrate, and they just climb these ladders moving around until they get to their appropriate location. The radial glia actually excrete various chemicals. And so if there's a specific neuron that's supposed to be part of the prefrontal cortex, for example, then it's following a specific chemical trail emitted by the radial glia so that it ends up in the prefrontal cortex. Where if another neuron is supposed to be part of the parietal lobe, then it follows a different chemical trail so that it ends up in the parietal lobe, and so on and so forth. These radial glia guide everything into their appropriate location. And there we have it. That is the end of this fairly brief chapter.